Good evening. Welcome, and I'm very excited this evening to talk about the microbiome and how gut bacteria affects many aspects of neurochemistry. And we're going to talk a little bit about mood, anxiety, and some of the information research on weight management. What I hope to do this evening is not only share the research, which has gotten a lot of press, from the New York Times to the Wall Street Journal to our academic articles, but really try to focus on some clinical applications that we can use in our practice. Larry Dossey, who's a brilliant writer, describes modern medicine as one of the most spiritually malnourished professions that ever existed. And in all the work that we try to do in integrative medicine is really to introduce some of that integrative spiritual as well as holistic treatment strategies, and understanding the gut and the microbiome is part of this. Thomas Insel, who's the director of NIMH, is quoted here a couple of years ago how the differences in our microbial world influence the development of brain behavior will be one of the great frontiers of clinical neuroscience in the next decade. He's a bright man, but he's also able to see the importance of the microbial world in our gut and its influence on brain and behavior. And that's what we're going to talk about today. The power of poop. We certainly understand and known for a long time how our gut health plays a role in many aspects of our health, from stress, anxiety, depression, to allergies. We'll be talking more about fecal transplants later, but this is something I received about a month ago in the news, in the journal, newsletter I get from Johns Hopkins, where fecal transplants, therapeutic poop is the name of the article, was effective for Clostridia difficile, and we'll be talking about this later in adults, but just to show you the power of the therapeutic poop. So we start with just some facts, but they're really fascinating facts that we tend to forget, and that 90% of the cells within your body are not human. They're bacteria and viruses. So we have 10 times more microorganisms living in our bodies than we do human cells. Tremendous amount of microbial bacteria. But what is, I think, more remarkable is not the number of cells, but the microbial genes number our genes 150 to 1. So even more dramatic than the number of cells, 10 times more microorganisms, but now we're talking about 150 to 1 genes, bacterial genes versus human genes. So we have about 22 to 25,000 human genes and over 3 million bacterial genes. And that is really going to be important as we discuss the microbial environment in the gut, and to understand that all these bacterial genes are producing proteins and enzymes that can have an effect on human digestion and human behavior. And then one of our core themes in all of our work in integrative medicine is the concept of biochemical individuality, and no two people are alike in any aspect, and we also do not share the same microbial makeup. So our bacteria in our gut and our skin throughout our body are probably most similar to our relatives and people living in the same environment, but they are not identical. When I was finishing up my training The most exciting part of graduation with my fellowship in child psychiatry was opening my own office and getting a fish tank. I was very interested in this symbiotic relationship and just loved watching the clownfish in the anemone. And this is kind of my visual image of what happens in our gut, but there are trillions of bacteria living inside our gut that are in this unique symbiotic relationship where we are providing protection and nutrients, and there's a vast number of roles the microbiome provides for us. And here's some of the established science. We know that the bacteria in our gut produces nutrients, regulates our immune system, our metabolism. But what we're now seeing in the literature is quite extensive, is that the gut bacteria influences neurotransmitters, affects our appetite and weight, can harvest calories and vitamins, and really can be responsible for cravings and our sense of fullness and satiety. I don't know how many of you remember the Horton Hears a Who, where there's a there's a small world of in Whoville, in this small speck of dust that Horton protects. And throughout the book, he talks about respecting these individuals. And it's kind of a theme that I share with my patients, where the understanding of the microbiome, even though you can't see them, that we need to both protect and understand, and part of our lifestyle and healthy choices are actually to nurture the microbiome in our gut. So I've used the term microbiome a few times, and that is the combined genetic material of all the organisms in our gut. We're all familiar with the concept of probiotics, which are microorganisms that are introduced into the body for beneficial qualities. Prebiotics, we've been hearing more and more about that term, which is food, non-digestible food ingredients that actually supports the growth of bacteria. And the theme that we're going to try to talk a little bit more about today is this concept of psychobiotics. And I kind of love the term. These are live organisms that, when ingested, produce a health benefit in individuals suffering from psychiatric illness or emotional distress. So we're going to try to focus on probiotics and healing the gut in an attempt to understand psychiatric illness. So we have these 4 to 5 pounds of bacteria in our gut, and there are many factors in our environment that decrease the bacteria in our gut. And here you see some of them: alcohol, antibiotics, pills, nutritional deficiencies, stress being the most common, steroid use, laxative abuse, infections, a whole host of things. But I think one of the things that we need to understand is that our bacteria flora starts from infants going through the birth canal. And that's where the microbial colonies are started, and so C-sections can be a problem. And what we find is certainly there's an increased rate of C-sections, and we are a number of examples how we're growing up with less microbial influences. So besides the C-sections, we grew up in homes of 5, 6, 10 kids. My grandfather tells me stories of 4 of his siblings sleeping in one bed. So everyone was closer, and there was more exchange of germs. We weren't drinking bottled water, and we didn't have hand sanitizers everywhere we went. The C-section research is actually quite extensive. The increased incidence of allergies, asthma, skin problems, celiac disease have all been related to the increase in C-sections. We also have found increase in eating disorders, particularly there's research looking at anorexia nervosa, C-sections causing increase in anorexia nervosa. In countries throughout the world, certainly in Asian countries and South America, C-sections are very, very much increasing as birth schedules are trying to accommodate the physicians and scheduling hospital time. So our obsession with C-sections as well, now our obsession with cleanliness has really, for many, had major implications in incidences of what we call inflammatory disease in people with depression. The bacteria that we were exposed to and that are part of our skin had very, very important parts of regulating our immune system. And some of the research published a couple of years ago looked at inflammation as a cause of depression, and the bacteria in our gut were one of the important factors keeping inflammation at bay. So the bacteria in our gut is so important that it's been referred to as the second brain. There are more nerves in the GI tract than the spinal cord. There are all the same neurotransmitters, and one in particular, serotonin, the vast majority is in the GI tract versus the central nervous system. We used to think of the how our emotional state affected our GI tract when we were nervous or anxious. Motility, secretions, and our appetite was affected. But now the research is showing that the bacteria influences our brain behavior and our ability to tolerate stress. There are lots of ways that the brain and the GI tract communicate. The vagus nerve has been demonstrated as the direct communication between the gut and the brain. So we have the ability of the brain to influence the intestine as well as the gut microbiota, and we have many changes based on the CNS. And then we also have this two-way street where the microbiota affects the brain. And today we'll talk about in detail some aspects of this two-way street where certainly mental health issues are involved in GI health, as well as obesity, appetite, and chronic GI disorders. The GI disorders is not what we're going to talk about, but obviously to mention it, if the gut flora is disrupted, we have inflammation, and inflammation can create a vast number of GI and metabolic problems. One of the true miracle advances in modern research in understanding the microbiome has been our ability to raise germ-free animals, mice and rats. And we've had this for over 50 years, where there's no intestinal microflora. And based on that, it's easy to do research, one, to understand what the role of the intestinal microflora are, but two, in developing treatments and understanding the immune response. But the most important data that I can share is that germ-free mice not only have no bacteria, but they have a very undeveloped immune system and an abnormal stress response. And this is from very, very old research. And we'll talk a little bit about how these germ-free bacteria has helped us uncover issues around weight. We're going to talk about stress tolerance and anxiety. And when we talk about weight, we'll start there. So the gut microbiome influences the energy balance, and we have more and more research that's demonstrating how important the gut bacteria is to the pathophysiology of obesity. And I think it's important to start with just some old history about what happens as we injected our farm animals with antibiotics. We know that low-dose antibiotics has produced for in all animals increased weight and more fat. And we are consuming those low-dose antibiotics. It was actually discovered by accident in 1948. A new antibiotic given to animals caused this dramatic increase in weight and growth, and some of the animals grew to weigh twice as much as the control groups. And ever since then, we've been injecting most of our animals with antibiotics. The vast number and the dollars that are spent utilizing antibiotics in our livestock and agriculture is really extensive. More than 50% of all the antibiotics developed in this country and used in this country are actually used for livestock. And 75% of those drugs aren't absorbed easily, and so they remain in the tissues and the muscles and meat that we ingest. The unabsorbed 75% are released into the environment annually, and this is seen in manure and fertilizer, in the water and soil that we eat. So this chronic exposure to low-dose antibiotics has really correlated with some of the rise in obesity that we have seen over the past 50 years. And many believe that this low-dose antibiotics, these residue antimicrobial agents, are contributing to this weight gain in our country. And there's some evidence to support that. So these are studies in mice, giving very tiny subtherapeutic antibiotic dosages, and there was increased adiposity, increased hormone levels related to metabolism, and increased fatty acid and metabolism of cholesterol and lipids. They essentially recreated metabolic syndrome and all the and the trajectory of weight gain by providing low-dose antibiotics. And then studies were done in infants, and these were 11,000 children born in the United Kingdom for a year. And they were given multiple courses of antibiotics at 3 different early life windows. And in the earliest time window, less than 6 months, there was a consistently higher body mass index later in life. So the antibiotics are used in a couple ways. One, we have research to support low-dose antibiotics, change the animal model with increased adiposity and fat. And we have a human model where low-dose antibiotics given to kids under 6 months of age, there was an increased body mass with these kids followed up almost to 2 years at 20 months. So historically, is that nationwide obesity prevalence seems to correlate with the antibiotic use in livestock. There's also research looking at increased obesity prevalence coinciding with the population's proximity to industrial farms. Again, many of these antibiotics are found in the water and the soil. So then we've taken this information on weight a couple steps further with research, again with germ-free mice. So there were no gut microbes. And when they were giving a fecal transplant, so the gut microbes of genetically obese mice. The mice bred to be obese, they had the increased ability to metabolize indigestible dietary carbohydrates that break down sugars and fats. And we'll talk about that in humans as well, but these particular strains of mice can break down these polysaccharides in our food supply that many individuals can't. So with this enhanced ability to break down carbohydrates into absorbable calories, if you will, they are more prone to obesity. So the nutrient value of food is not just on what's ingested, but what is the ability of our gut microbes to metabolize particularly the carbohydrates in our food supply. So when we look at human studies, one of the things that seem to be most important is something called diversity and bacterial richness. So this was a study with Danish individuals, 123 non-obese and 169 obese. And those individuals that had the lower bacterial richness gained the most weight. So low bacterial richness means the least number and variety of bacteria in the gut, increased adiposity, insulin resistance, increased lipids, and this was compared to those that had high bacterial richness. We had also, a very, very similar thing has been found with anorexia. Anorexic patients also have very, very low bacterial richness in some of the studies that were done with patients with eating disorders. This is just a slide to share with you how much research has been done, and we could fill 3 or 4 more slides looking at the gut bacteria and its relationship to weight, weight gain, and some with food craving. So there's a lot of literature there. There are just a few additional examples that I think are important. This is a study of H. pylori, and I think it got a lot of press in terms of originally thought to cause ulcers, but in reality, H. pylori is a very critically important bacteria that regulates acid in the stomach and also regulates the hormone ghrelin. And ghrelin is a postprandial hormone that tells the brain that you're full. So after you eat, the ghrelin goes down. And H. pylori has a very important role in decreasing this postprandial ghrelin. So this was a study looking at 92 veterans, and those that were treated with antibiotics to eliminate H. pylori gained more weight in comparison to the peers that weren't treated. And here's an example. 50 years ago, 80% of Americans had H. pylori, and now with our 6% of American children test positive for H. pylori. So let's just try to understand it. So the ghrelin is a hormone that causes you to be hungry. So think of ghrelin and think of growling because you're hungry. So if we go back, that H. pylori decreases that postprandial ghrelin. Okay, so that's important. So ghrelin goes down, you're not hungry. Without H. pylori, you can't regulate that appetite satiety, and you will continue to be hungry and eat and gain weight. This is another bacteria, Bacteria theta, that has some of those enzymes we were talking about in the mice model, where there's, for this bacteria, 260 enzymes breaking down large complex carbohydrates that help digest these complicated plant foods into simple sugars. Now, we don't have most of these enzymes, so these polysaccharides would just be undigested and pass through us. But our gut bacteria, the Bacteroides, will digest and ferment these polysaccharides, generating cholesterol and fatty acids. And we've also shown that some of the sugar-free, low-cal artificial sweeteners also affect the microbiome. So rodents that don't have this Bacteroides have to eat 30% more calories to gain the same amount of weight. So it's pretty significant difference. Now, in terms of weight, we also know malnutrition is another significant variable, and starvation causes the stomach to increase in acidity, there's distension, there's bloating, and this changes the environment which bacteria can live. And again, testing our patients who have restrictive anorexia, they have very abnormal gut bacteria. A group of, there's been very little research. There's some research going on at University of North Carolina on anorexics and the gut microbe. This was one on children with malnutrition in Bangladesh. And these were monthly stool samples collected. Children were compared with malnourished kids before and after they received their food and nutritional interventions. Compared to the healthy kids, those severely malnourished children were found to have immature microbiome, both at the start and the end of treatment. And those with severe acute malnutrition, this immaturity in the gut persisted despite these nutritional interventions. Again, there's an important window early on to see the microbiome. There's now research looking at how the microbiome actually is controlling many aspects of our diet, where the bacteria is looking for their own survival. Remember the 150 to 1 number of genes. So the bacteria is looking for the sugars or the refined foods to feed their growth. And some of you may be familiar with candida and yeast, but certainly there's high sugar cravings in those individuals who have yeast. So whenever there's any information on how something might affect weight or appetite, somebody writes a book, and this is just the beginning, and now we have books on the microbiome diet. It's by a gastroenterologist who essentially is taking some of the research that I shared with you and talking about eating fermented foods and probiotics, and that will cause you to lose weight. There's actually a couple other books that have come out with a similar theme in the past 6 months. I think it's all important information in terms of taking care of one's health. I think the research is not quite ready for a simple weight loss program by simply eating more fermented foods. I do think that weight management, restoring weight for anorexics, or losing weight for those with binge eating or overweight, that the microbiome is a critical factor in weight management. It's not the only answer. So I want to talk now a little bit about probiotics in mental health treatment. It was 1910 where it was first reported that live bacteria improved symptoms of depression in adults with melancholia. And then 1923, acidophilus milk was recommended for the treatment of mental illness, psychosis, as being better than any other physical treatments that they had at the time. We know that probiotics administered to animals has dramatic effects on neurotransmitters in the brain. These are just some of them, but oral administration of probiotics increased the plasma tryptophan, decreased serotonin metabolites, so essentially increased serotonin, and increased dopamine. There's also literature we'll talk about where is acetylcholine and Gaba and all the other neurotransmitters that we think about in optimal brain function. The probiotics have been shown to affect these chemicals. This is one study of 36 women who were giving, split in 3 groups. One was giving a fermented product with probiotics. And what they were able to show, looking at functional MRI scans, so looking at the brain in action, if you will, that after a 4-week intake of this probiotics, the activity of brain regions that control central processing of emotion was clearly statistically significantly different for those that didn't get the probiotics. So a major difference seen, and here's just the visual of the fMRI studies. Here's some studies on rats and personality, 8-week-old mice, again, that had no intestinal flora. These were germ-free mice. What was interesting is the researchers transplanted the microbes from young mice that were timid into the guts of mice who were more risk takers. And the timid mice became outgoing, and the outgoing mice became timid. This was done a couple of years ago, but the microbiome, the gut bacteria controlling personality in mice. So here we have the other thing that I shared with you, how probiotics can affect tryptophan, and tryptophan is necessary for the production of serotonin, and obviously most of our medications in psychiatry are based on trying to raise levels of serotonin. So if there are ways that we can raise tryptophan through diet, supplement, or supporting the microbiome, we would increase serotonin as well as melatonin, which is made from serotonin. The stress research has been quite significant. We've known for years that stress can influence our microbiome and our GI tract. When the brain senses stress, that there's many chemical responses that affect the bacteria in the gut. Here you see just a visual of two different GI tracts. The one on your left, where this bad bacteria can damage the intestine, causing we call that leaky gut syndrome, poor absorption of nutrients, and as the bacteria is now protecting the GI tract and allowing for enhanced nutrient absorption. We also have literature showing that norepinephrine levels under stress. So the increase in norepinephrine in the intestine stimulates a range of bacterial species, including E. coli and a Campylobacter species, and these both can cause infections. So I shared with you early on that part of the syndrome of germ-free mice, those mice without any microbiome, that they have an abnormal stress response. So they have increased activity what we call the HPA, the hypothalamic pituitary adrenal axis. And what the research has been showing and repeated is by adding the probiotics, in this study bifidobacteria, they were able to maintain a normal stress response. So adding probiotics to rodents that had an abnormal stress response, they were able to normalize that response. And to me, this was a fascinating follow-up because at different ages of probiotic treatment, there was a different response. So here, germ-free mice with this poor stress response, and they were giving restrained stress, and they were stressed out. So the researchers looked for their rise in cortisone, and if they gave the germ-free mice repletion of the gut microbiome at 6 weeks of age, which is considered adolescent in mice, there was a normalization of that stress response. If they waited 2 weeks after that, so if the mice were colonized at 8 weeks, the altered stress response was persistent through adulthood. And again, very important as we try to think about prevention of major psychiatric illnesses. And one thing that is really amazing to me is all major psychiatric illnesses are evident by age 14 in the vast majority of individuals. So if we begin to start understanding and protecting and repleting bacteria in our young adolescents, we'll be able to begin to think about models of prevention. So here, more research. This was a single strain of a bifidobacterium elevating tryptophan. And we talked earlier about inflammation and depression. And what this study also was able to demonstrate is that probiotics could decrease those inflammatory markers, the cytokines, oxidative stress, and other inflammatory markers. Again, jumping around to a human study, this was 39 patients with chronic fatigue, and they were receiving a placebo or 24 billion CFUs of lactobacillus. And this was interesting because not only there was an increase in lactobacillus, there was a decrease in anxiety among those taking the probiotic, but there was also an increase in the bifidobacteria, even though they were not giving extra bifidobacteria to these individuals with chronic fatigue syndrome. So this is one of many studies showing that supplementation with one strain can support the growth of other health-promoting species in the intestine. Here's some more stress management and studies that were done in mice. They were given Lactobacillus rhamnosus, which is the brand in Culturelle. And here they were able to show in the maze test that those that had the probiotic broth were more relaxed, able to explore open space twice as much as those non-bacteria fed mice. So here, a very well done study looking at ingestion of probiotics resulted in lower stress in this maze test and lower level of the stress hormone cortisol. And this was very significant, a probiotic being able to lower cortisol. I think many of you are familiar with Gaba, which is a main inhibitory neurotransmitter in the brain. It promotes relaxation, inhibition, so decreases anxiety, and important for maintaining healthy sleep. So again, a study with the Lactobacillus rhamnosus that these probiotic was able to modulate the GABA receptor subunit and actually promote a normal stress response and positive mood changes. increased expression of Gaba receptors with probiotics. And here's some of my favorite proteins, is that in our germ-free mice, we have lower levels of a compound called Bdnf. It's brain-derived neurotrophic factor. And this is one of the important markers we have for a healthy brain, a happy brain. So it's a protein that's associated with learning, memory, and positive mood. It's really seen as neuroprotective, and it is increased when we exercise, we eat well, and we're stimulating our brain, and stress decreases Bdnf. Bdnf is a zinc-dependent enzyme, so zinc deficiency also decreases Bdnf. So it's a very, very important marker of health. I call it the miracle grow for brain cells. But here, what we can see is providing probiotics. Healthy bacteria is actually able to stimulate the synthesis of Bdnf. So that's our miracle growth for the brain. You see the nerve cell on the left. We have stress, inflammation causing decreased Bdnf. But adding probiotics, you get this dramatic enhancement of cellular function. The cell grows bigger, dendritic branching, and the clinical symptoms are mood, memory, and there's a huge neuroprotective function of Bdnf. It's a really dramatic literature, mostly in animals, but adding probiotics was able to stimulate such a neuroprotective chemical as Bdnf. These are some small studies, but continuing to point to the importance of Acidophilus and probiotics. It was a double-blind controlled trial. These were healthy individuals, mean age of 61, and they consumed a yogurt drink or a placebo for only 3 weeks. And just on simple rating scales, those who consumed the yogurt drink had, those that had rated their mood as poor reported feeling happier. So it wasn't a happy pill. It wasn't a cocaine or a drug-like effect, but those that had rated their mood as poor reported feeling better. And this was double-blind, placebo-controlled. Everyone was taking the same food. Similar to what I shared with you earlier, if we put rats under stress, we have this inflammatory response. We can decrease that inflammatory response, increase concentration of tryptophan by adding strains of probiotics. Here's one of the studies we talked about earlier, looking at dysfunctional MRI, individuals given placebo or probiotic. The probiotic was associated with changes in blood flow to brain regions involved in central processing of emotion and sensation. And we talked about that. And even memory. Again, this was the mechanism with Bdnf, but another probiotic, a study looking at levels of acetylcholine, and those that received the probiotic, there was Bdnf expression and improved memory. So it was a lot of examples. I apologize, they were all different, but I wanted to give you a flavor for what's happening in the literature. And lots have changed. We've gone from, thinking that bacteria were evil, chemicals, evil microbes that were going to destroy us, to now understanding how important they are to our health. We talked about the therapeutic proof. I want to focus now on something that hopefully many of you are familiar with, but I wanted to bring it to some clinical reality. And what I spent the first half of the evening talking about is how important the microbiome is to all the neurotransmitters in the brain, and it is a lot of diverse research. But we actually have a mechanism to measure one chemical based on dysbiotic strain of Clostridia in the gut, and many of you are familiar with HPHPA. So this is a metabolite of phenylalanine produced by a number of Clostridia species in the intestine. And what is critically important is that HPHPA is similar to dopamine and norepinephrine, and it affects the metabolism of these neurotransmitters in the brain. And we've seen patients with depression, anxiety, OCD, autism, and schizophrenia that have very, very abnormal levels of HPHPA. So we talked about the 4 or 5 pounds of bacteria. Well, there's 100 strains of Clostridia, and there are a few strains that produce this HPHPA in the intestine. The HPHPA is absorbed and filtered through the kidney, and we can detect it in the urine. And what this chemical does is it inhibits the enzyme dopamine beta hydroxylase, which is the conversion of dopamine to norepinephrine. So we get this buildup of dopamine. And buildup of dopamine can feel very irritating, anxiety-provoking, and people don't feel well. To me, it's amazing the tremendous range of symptoms. And again, it's been everything from anxiety to just agitation to even psychosis with elevated levels of HPHPA. What is also interesting is what happens with HPHPA. We get this buildup of dopamine, but we also have this unique ability to break down dopamine, and that's based on the COMT enzyme. So it's one of the ways that dopamine is broken down in the synapse. And if we have different variants of this enzyme, what we call different polymorphisms, the HPHPA in our system can be even more dramatic. So if these are 3 variants, you see here 2, val-val, met-met, and the normal is met-val. And these variants affect how efficiently the COMT gene breaks down dopamine. So with the met-met variant, you break down dopamine very slowly, so you have higher levels of dopamine in the synapse, which if you had HPHPA, which means you're continuing to not break down dopamine to make norepinephrine, you have a more enhanced, I believe, mental health picture with increased agitation and often anxiety. These are just some examples of the elevations of HPHPA. This is a gentleman with OCD, and this is a gentleman with kind of an agitated depression. So in summary, the HPHPA inhibits a key enzyme that converts dopamine to norepinephrine. These dopamine metabolites use up glutathione and makes us more susceptible to some of the toxic chemicals in our environment, from heavy metals to pesticides, and sets up accumulation of these toxins. Our standard American diet, the junk food, if you will, also coincides with inflammation and a profound effect on our microbiome. And what I want to end with tonight is the concept of fecal transplants, which has really revolutionized the treatment of Clostridia difficile in this country. But it's been around since the 4th century in China, in 1958, treating some patients, and recently approved by the FDA as an experimental drug. The fecal transplants are done a number of ways: enema, is the most common or suppositories. There was a group at MIT who recorded all their stool samples in test tubes for an entire year, plus recorded everything they ate to try to monitor the microbiome. This is a gastroenterologist in Boston who had to mix her own stool samples by using a blender from Target and transplant it into the patient's colon because it was so effective for her for the treatment of Clostridia difficile. In 2013, the randomized controlled trial had to be stopped by the New England Journal of Medicine because they said it was unethical to continue to deny the transplants to the control group because it was so successful in the treatment of this potentially life-threatening GI illness that is very hard to treat with antibiotics. So we now have stool banks all around the country and primarily focused on treatment of Clostridia difficile. The reason that I wanted to kind of share this with you is that, it just convinced that our gastroenterologists are way ahead of the curve, and I believe soon that the research is becoming so significant that we might be looking at this for mental health problems. Fecal transplants to introduce healthy flora to those struggling with mental health issues. Here's the wonder cure with the fecal transplant. So, in summary, we kind of need to ask the question, who are we? I've said it many, many times, the microbial genes outnumber us 150 to 1. So there is lots of genetic material in our GI tract making protein and enzymes for the sole survival of this bacterial community, not for our survival. So oftentimes we need to think that our fate might be more in the hands of our microbiome than our own genes. I think what's clear is microbial diversity is an important part of health, as well as now weight and stress response and depression and anxiety. Maintaining a healthy GI tract is critical, and looking at tests like the organic acid where we have a treatment marker that could be easily assessed and treated aggressively. Most of the time we're using high-dose probiotics. I use 50 to 100 billion for elevations of HPA. Oftentimes we need to use antibiotics, and there we would be looking at flagyl or vancomycin, different theories as to how to treat it. Most people are using a pulse method with alternating 1 or 2 days on the antibiotic and then a couple days off. I have found patients who have responded, particularly younger patients, to just probiotics. Sometimes we need to use the antibiotic as well. Whenever we talk about integrated medicine, we always come back to the concept of biochemical individuality. Our microbiome is unique, and our ability to support our microbiome needs to understand that uniqueness. We have even gone to find that linked chocolate to specific chemical signatures that might be programmed into our metabolic system by guess who? Bacteria in our gut. So they were able to demonstrate in this research paper a number of years ago that the activity of gut microbes in the chocolate lovers were distinctly different from those individuals that were not as obsessed, if you will, or craved chocolate. So this is a recent editorial in the New York Times. Microbiome might be looking out for itself, and the importance of this critical major influence on our behavior. So in summary, we have many major neuropsychiatric illnesses, and we throw many medications at our patients looking for an effective relief of symptoms without understanding anything to do with the cause. And sometimes looking at the gut, treating with probiotics, even if testing is normal, they're simple, they're safe, they're not expensive, that we begin to see some improvements in behavior. So, I term this a simple approach for complex puzzle. Neuropsychiatric illness and mental illness is not simple, it's complicated. Family dynamics, individual dynamics, and genetic biochemical dynamics are all interplay. But the brain-gut kind of bidirectional role in creating and helping evolve both the immune system, the endocrine system, and the brain makes us want to think twice about looking at supplementation. And supplementation is simple with probiotics that can be easily obtained. Share a story in closing. I've written a number of articles on individuals with OCD that had elevations of HPA, and a gentleman who had long-standing trichotillomania for, I believe, almost 30 years, read the article, and he surmised that OCD has compulsive behaviors, and his chronic hair pulling is compulsive. So we started taking high-dose probiotics without any testing, and within a number of months, he was able to stop his trichotillomania, which is chronic hair pulling, and within 6 months, he wrote about his success in the treatment of this compulsive illness, that we have very few medications and or therapies that consistently work. So friends, with benefits, 100 trillion bacteria we didn't know we had. There is progress in depression and eating disorders, and it's clear that there's a relationship between this gut microbiome and brain function. As we understand this better, we might be able to target specific strains for specific illnesses. I know there are supplement companies that are recommending this strain for depression, this strain for weight management. I have not found those to be consistently helpful. I use a multi-dose strain of 50 to 100 billion CFUs per day. So the question is, which species are in your feces? And in all the books that we've looked at, we've tried to help people understand the importance of the microbiome in psychiatric illness. Again, thank you for your time, and I appreciate your listening this evening.